All right, in the interest of time and making sure we have enough time for everything and that numbers are starting to steady out, we'll get started. This is being recorded and will be available later for those who maybe had to log in a little late. Welcome to our next webinar. This one focused on the biomanufacturing readiness levels hosted obviously by NIIMBL. I'm going to help to co-chair this session. My name is Chris Roberts. I'm the Associate Institute Director with NIIMBL. Most of the presentation is really going to be run by Sandeep Kedia, who I'll introduce in a bit. Maybe we can go to the next slide. All right, so a bit of some announcements, some housekeeping. So for those who have logged in, you're muted throughout the presentation. That's an automatic function of the way the webinar is set up. So please use the chat feature to ask a question or post a comment. That should come to all panelists. I'll be primarily the one monitoring that while Sandeep's giving the main portion of the webinar. This is open to NIIMBL members and non-members. This is not a NIIMBL-only event, so please do not post NIIMBL confidential or proprietary information in the chat or Q&A. There is a survey that you'll receive a link to by email soon after the webinar. Please complete that survey, and upon doing that, you'll get immediate access to the webinar recording. We value that feedback, independent of getting access to the webinar recording. We highly value your feedback on all the aspects that are going to be covered there, and we use that to help for continuous improvement as we work through all of our processes and communications. Next slide, please. So because this is both for NIIMBL members and non-members, we're going to do just a couple of slides here where I'll cover some background on what NIIMBL is about, for just to help orient, and then it sets a stage for what then Sandeep is going to cover. So we are formerly the title would be we're a public-private partnership, which some people just think of instead of in terms of a consortium, but it's a mix of different partners. So we have federal agencies. Our sponsor is NIST, but there are other federal agencies involved. We have large and small companies who are members. They can be manufacturers, vendors, technology developers, others. We have academic institutions. We have not-for-profits of other sorts, and even we get some support from states. We're dedicated to advancing biopharmaceutical manufacturing, innovation, and workforce development. Those are intimately tied. We're one of 16 institutes within the Manufacturing USA network, and with more institutes likely to come. And they are synergistic, but typically non-overlapping institutes. But all of the Manufacturing USA institutes have a common goal of driving and advancing and maintaining US leadership in advanced manufacturing. So next slide, please. I'm not going to read this off verbatim. You can read it for yourself, but our mission formally is in the biopharmaceutical manufacturing space. It supports not only innovation of technologies, but also standards and maintaining and developing and expanding a world-leading biopharmaceutical manufacturing workforce, and ultimately advancing US competitiveness. So what do we do? At a very high level, we work to try and develop and de-risk transformative technologies in the manufacturing space for biopharmaceutical manufacturing and the related processes to help expand advanced manufacturing capability in the US. Although that, of course, does have worldwide implications, because many of our members are international companies, especially the large companies. So we look to solve pre-competitive industry-wide problems that give the US an economic and security advantages. We are funded primarily through the US government. We share best practices that promote regulatory harmonization. We look to enhance accessibility, potency, safety, efficacy of biopharmaceuticals, obviously for the US population, but that does have advantages worldwide as well. And we look to support best-in-class education to build the workforce. So next slide, please. So this is my last one before I'm going to hand over to Sandeep. So what we've found historically, or not we have found, but it has been found historically, and NIIMBL certainly has seen this as well, is that at least U.S. investment, and I would say other federal investments from other countries, has been extensive and quite effective in funding things at, maybe you want to move your cursor over to that one to the far left there to show the lower, what we call manufacturing innovation readiness. So these would be more research, early stage development types of projects, development of new ideas, maybe even spin-outs into small companies. But then there tends to have been a gap over many years now for seeing those make their way all the way over to the far right, which is where the private sector, primarily your manufacturers, your suppliers, what have you, are then actually commercializing these technologies. And so there's a concept that Sandeep's going to talk about in detail around readiness levels. And for us, being NIIMBL and the biopharmaceutical manufacturing community, there you can think of it in terms of manufacturing readiness levels, but it's a bit different for different industries. And so what we're talking about today is really focused on helping to understand how you move through those different readiness levels, how they help one to move through that middle gap, which is where NIIMBL is tasked to help the community develop new technologies, educational components in order to help bridge that gap. I think maybe in the interest of time, I'll stop there. We could move to the next slide and then I can introduce Sandeep. All right, thank you. So sorry, my screen just froze there. So here we go. So Sandeep currently serves as a senior technology fellow for us here at NIIMBL, where he led the efforts to develop this updated BRL framework, biomanufacturing readiness level, or biopharmaceutical manufacturing readiness level, and supports NIIMBL-led programs in his other roles, such as the big data program, vector manufacturing. He helps support our technical advisory committee, among many other things. He joined NIIMBL in 2020 after spending over 20 years in the biopharmaceutical industry, where he led CMC programs that span from development to commercialization to bring a number of medicines to the market. Most recently, before he joined NIIMBL, Sandeep was a director of product development and head of CMC at Sobi. He also led the process development group at Alkermes and at GSK, among other companies. And today he's going to share highlights of the BRL framework for technology adoption in biopharmaceutical manufacturing. All right, so thank you for your time, everyone. And Sandeep, over to you.
Hi, everyone. Thank you, Chris, for that introduction. So today's session is going to be on biomanufacturing readiness level. And this is a more about information sharing sessions, and basically would like to get your feedback. So what I'm going to cover in this session is why do we need this framework, the purpose of BRL, the framework itself. I'm going to cover a couple of examples and then share with you on how you can give your feedback. So first, I would like to point out, this is an uphill task that was undertaken by NIIMBL. And I would like to thank all my co-authors, who you can see listed here, come from various industry, academia, and federal government agencies. We published a BRL framework document as an open access in biotech and bioengineering. And basically, you can look at those publications. You can download those and get more information about that. One key disclaimer I would like to point out, as you see some of the federal folks, ex-federal folks who are part of NIIMBL now, this only represents the view of NIIMBL and should not be construed to represent FDA's views or policies for technology adoption in biopharmaceutical manufacturing. So we know that MRLs were initially developed by DoD, and they're well-established tools for assessment of government-sponsored programs. What MRL is not always helpful in biopharmaceutical industry because biopharma is highly regulated, as we know, through global authorities. It's a high pain of failure from a clinical perspective as throughout the life cycle of technical advocacy. There is a need to demonstrate control of both process and product in order to manage the residual uncertainties. Because as a complex biopharmaceutical systems, the meeting product spec is necessary, but not sufficient. Commercial biopharma development and lifecycle management is based on end in mind, rather than the sequential milestone-based thinking. So I think, combining some of those challenges that the MRL doesn't address, we in NIIMBL bring together private, public, federal, nonprofit, academic stakeholders to accelerate development of innovative technologies in biopharma, as Chris mentioned. So these BRL will lay out a groundwork for a shared vocabulary for assessment of technology maturity, readiness levels in commercial biomanufacturing, effectively meets the needs for this critical, specialized, and highly regulated industry. Hence, we basically developed this BRL framework. So the purpose of the BRL framework is to create a landscape for technology adoption, right? So we're thinking from anywhere from ideation phase all the way to commercial process and implementation of those. So what we would like to establish at the end of the call here in the webinar is, present to you that we created a shared vocabulary we can going forward. We have an assessment toolkit that we can commonly use, a value proposition that is clearly stated out and understood with the end in mind. So the end in mind is the adoption of the technology that's been in development. So now I'm going to describe, the framework on what went into creating this framework. So first is the value proposition. For any technology that we want to make sure, we understand the values, the likelihood of technical success, right? How key is the technology, the assay, the method in terms of technical success? Now, not only thinking from a laboratory perspective, but all the end in mind. So it could be a commercial implementation. The likelihood of deployment, and this is a key thing where, we have to kind of understand about the stakeholders management. So the three main categories are, from a concept development perspective, it's basically developing from innovation, ideation stage, all the way to proof of concept. And we have broken down into BRL 1, 2, and 3, which you'll further see on how we elaborate those further. The second phase is the concept demonstration phase. And this is typically done in the pilot operations or pilot ops. And this is actually taking the proof of concept for a technology or methods or assays and demonstrating for a relevant biopharmaceutical application, and such as implementing in clinical trials, things like that. And concept final realization. So this is basically, the end product for the technology development folks. Where basically we are now finally implementing the technology or the methods of the assays for our commercial biopharmaceutical products and making it better. Now, what went in, right? So the hurdles, technical readiness. We want to make sure our technology is ready and can be ready throughout the course of the BRLs from 1 to 9. So we thought about, feasibility, like scale up, process controls, process capability, the CP, vendor management, materials management, thinking about what are basically materials availability challenges that we foresee going forward, not just making one instrument, but making it all the way through commercial scale and supplying at a commercial scale. From a quality readiness perspective, what we thought about, putting a quality management systems plan. Do we have a plan in place and it's ready? End-to-end cost model for the technology. Some key process parameters or validation master plan, ICH guidelines, which are well described on how to implement these things, and process of change controls throughout the facility. In terms of operational readiness, some of the key thoughts that we need to think about is stakeholder management. Who are the people that need to be involved in decision-making for the application to the end product? About facility capability, what if there is any modification that is required. Thinking about workforce development, right? This is often understated a lot. Do we have the trained workforce for those technologies or assays or methods? And it can cause supply chain disruption from existing processes. So, thinking about those up front and to ensure that we have operational readiness going forward. So what we did was we categorized those and we create some subcategories so we can combine those. So from technical readiness, we're thinking about technology from an unmet need. And this technology is our technology ready, essentially? And can that be implemented throughout the life cycle of the technology or the methods? From a process control and robustness, we need to answer questions around how robust the technology is with respect to the process, and in terms of controls, what is required, and can that be put in place in the facilities. From a material identification, making sure, we have more than one vendors who can supply the raw materials, for instance, or we have abundance of those raw materials availability that we don't run into issues of supply chain later on through the life cycle of the technology. From a quality readiness, definitely understanding the value proposition and clearly defining it. Your value proposition would change as it goes on and it basically matures throughout the life cycle of technology. Thinking and implementing quality management systems which are well designed from ICH guidelines and implementing during the development of the technology, right? And creating a quality management plan in place. In terms of operational readiness, things like business fit, stakeholders management, right? What are the questions that we need to answer to make sure the technology can be readily adopted or implemented in a facility? Thinking from a facility perspective, the changes, the modifications that would need to be made, or in terms of workforce, we need to think about the training that needs to be involved, systems that needs to be created, things like that. So these kind of form the 3 top categories and the 9 subcategories for this framework. And what we did was we basically combined the phases and the categories and the subcategories to develop the framework. So that you have essentially 9 phases going across the top row, BRL 1 through 9, and they're divided into concept development, demonstration, and realization. And then you have categories such as technical readiness, quality readiness, operational readiness, which is then subdivided into the various subcategories that we need to address at some point in time. Overall, what you'll find in the publication that I mentioned earlier, there are essentially over 150 activities that goes across the 9 phases and 9 subcategories. Now, it may sound a lot, but we basically hence divided into a phase appropriate advancement for those questions to be answered. And some of the questions may not be applicable to your technology or development phase, basically, right? So, but most of them hopefully would kind of stir up the questions that need to be answered at some point in time. So they are basically mentioned in Table A for the BRL framework document. And how does NIIMBL come in play? So, once we develop this framework, basically we understand, from a concept development, those are basically getting a technology from ideation stage to proof of concept. So those we qualify as pre-NIIMBL activities, right? And then when it comes in play from a concept demonstration space and the concept realization space, we say it's a NIIMBL activity. And the way it works is NIIMBL plays a key role in communicating the attributes required for transport across the BRL3 to BRL4 phase. This is a very key membrane right now. Thinking about, once you acquire the proof of concept in the laboratory settings, how do you actually start implementing and developing your technology or tools to be finally implemented in the commercial space? Also, you can think about NIIMBL as a transporter, a co-transporter of protein, that facility that transports from BRL7 to a commercial space. Now, BRL7 is the pilot operation space, transporting that to a commercial space. So, our mission is to enable deployment of modern technology in the manufacturing settings. So we help basically go across the space from a pilot ops to commercial space. Thanks, Chris, for that note. And also in the commercial setting, you may have a mature technology that could be implemented across modalities or across products, existing commercial products. And NIIMBL can basically play a role in essentially establishing high-value product settings and what are the required criteria or attributes for that to deploy across the BRL8 to BRL9 space. So what is the various levels the BRL levels consist of, and what are the criteria and summary exit criteria for those each phases? So we'll focus on phase by phase and we'll go through the things. So for BRL1 is basically an ideation phase, like I mentioned, it's a concept ideation phase. And in this phase, what you do is a concept is conceived, the idea is conceived, and based on solid, generally recognized scientific and technical principles, right? Data is available, preliminary data is. In BRL2, it's formulation of that concept. We document description for the technology and address of feasibility and benefit. In BRL3, we establish proof of concept. And this is basically where we publish documents, we publish papers on the proof of concept, showing one technology is better than the other, in the state of the art, things like that. And this publication, this could be a publication in a peer-reviewed journal or a disclosure or a patent or an in-house technical review with key supporting data. Now we go from a development to concept demonstration phase. Now we are talking about the pilot scale activities. And this is where we think about GMP space. So we need to understand in BRL4, it's pilot design 1, where we are trying to build a prototype. So we document the test performance with the industry relevant feedstock, demonstrating agreement with the expectation. And this is a preliminary plan developing for scale-up integration robustness requirement. So as you move along the phases, you'll see each of the phases actually reminding you things to be addressed for the next levels. In the BRL5, which is predominantly the scale-up integration and limited robustness scale, which is we identified as pilot design 2 phase, in this you demonstrate the results showing technology meets the acceptance criteria, determined in the areas of manufacturing, scale-up, integration, and robustness. The determination that means instrument components supporting technology, such as software, can be reliably produced and commercially available. So, aspects that we need to think about and quite often we may oversee. The next phase is the BRL6, which is the development of drug candidate. Now here we're actually testing our technology or methods with an active drug candidate. So technology is used for early GMP supplies for active drug biological candidates, a second generation process for licensing compounds. So essentially you're bringing an old historical process back into force, using the new technology or the new methods. In BRL7, we want to ensure that our technology is now ready and GMP systems are ready and built. So this is where we demonstrate technology readiness to manufacture pivotal batches. And pivotal batches, it could be phase 2B, phase 3, or bridging studies for approved IND. So those are the aspects, we're talking about essentially entering the GMP space. In the concept realization, where we talk about BRL8, this is where the system is validated and approved by FDA. So these are FDA, EMA, PMDA, other regulatory agencies across the globe, essentially. So this is commercial operations. This is where our technology is used in at least one licensed demonstrated commercial product. So we're demonstrating a technology for one licensed commercial product. And in BRL9, finally, our technology is demonstrated in routine commercial manufacturing of multiple approved products or multiple companies or multiple sites. So this is now a broader implementation of established technology through successful commercialization. So we call it the tech deployment. So these are the 9 key established criteria and some of the aspects of the summary of the key exit criteria that would be required to move along the phases of development for the technology. So I'm going to basically go over a few examples here to give you a little bit more on how we actually use this process. And it is giving you a different flavor. So obviously this does not cover all the aspects, but how we actually plan to use the framework. So this is about developing a protein tag, right? Affinity tags that's been developed in the university incubator. This is improves the original protein fusion compared to the protein A or LAC-Z that are commonly used. So the person going through the table, also through the table A, which is more extensive and gives a little bit more criteria in terms of understanding and answering those questions. So in this ideation phase, yes, the person has developed the removal protein tag, identified and published in a peer-reviewed journal based on the work in the university incubator. In BRL2, yes, the concept is formulated. Current method of protein A is shown improvements in DEF compared to LAC-Z, or protein A, and it's demonstrated off the shelf and nomination solutions. In terms of the proof of concept established, it's been demonstrated in the lab in a number of proteins and purification conditions in laboratory settings. So yes, this task has achieved the proof of concept. From a BRL4 perspective, prototype built, laboratory methods and SOP are being developed for biomanufacturing drug candidates. This is where, we state the technology is currently established in BRL3 and is being developed to establish procedures and method to accomplish BRL4 as per appendix. So how do we evaluate that, right? So when I mentioned over 150 activities across the 9 phases and 9 subcategories, I have listed out some of them, key things here based on the previous examples. And this is to give you some idea on how you can use the framework. So for instance, technology for the unmet need could be in the BRL4 space. Yes, it's demonstrated proof of concept. It's been developed to use for drug candidates, etc. Right? But from a process robustness and control perspective, we still need to validate performance for the method development. So that's ongoing. From a materials perspective, component specifications established to reliably produce it commercially. So those things are ongoing at the moment. And you can see some of the idea here is, they're not all in BRL3 buckets. Some of the components for the subcategories can be in any bucket based on what those bucket relays and where the development for the technology is. From a quality readiness perspective, from value proposition, they're developing cost models for materials in manufacturing settings. From a QMS perspective, they might be at the level where identifying suitability requirements per ICH guidelines. They are still developing their quality management systems plans in place. From an operational perspective, from a business fit, workforce fit, they're training personnel for the method development and transfer. From evaluation of technology adoption, the workflow requirements are basically being worked on, and they could basically rely on between the barrels of BRL3 and BRL4. So you can clearly see, not everything needs to fall in one category. You can work on multiple categories based on where the technology is and how this is going to be used. This is another example for the glycoform analysis, and this is basically to develop this was developing a new structure assay that had been developed by using high-res mass spec coupled with specialized HPLC techniques to yield the rapid glycoform analysis compared to this gold standard QC techniques for released glycan that we currently use for existing drug candidate. So in this example, yes, the new assay has been identified for glycoform analysis. It basically covers the key major glycoforms of interest for development. It is faster technique compared to the existing technique by X-fold, and it's published in Y articles, so it could be basically internal articles as well. So from a concept formulation perspective, the assay was developed and optimized for a drug candidate. From proof concept, the proof concept was demonstrated for the new assay. The assay method developed for new drug candidate for glycoform analysis. So this is where they identified, various glycoforms of interest for the drug candidate, and they were able to basically identify and label it. From a BRL5 perspective, new assay used for identification of glycoform during the development of new clinical trial materials. Method yielded a satisfactory measurement compared to the existing analytical technique. So this is where we are comparing the gold standard versus the new technique. From a BRL6 perspective, the drug candidate currently is being used to develop in the new drug candidate at contract lab for clinical trials. There have been plans for bridging studies between the release method and the new assay method, and assay development is currently deemed at BRL5 level, progressing to achieve BRL6 as listed out in Table A. So again, going across the table, around 150 activities on how this was demonstrated. So, tech demonstration underway for a clinical drug candidate. So, you can clearly see where technology is currently. There are plans and bridging studies are underway as per ICH requirements. From a quality readiness perspective, the system validation studies as per ICH requirements are underway, and system sensitivity analysis completed from lab to pilot. So, this is basically some of the value proposition questions coming in way. From an operational readiness perspective, developing the risk register to implement the technology across the drug development candidate for this one. And there are plans for tech adoption and site transfer ongoing, since this was basically developed at originating site and then transferred to CDMO, essentially. So those plans are underway. So as you can clearly see, this is basically a technology on various scales that have been laid out with various subcategories and where they actually are on the spectrum. This is the third example. This is basically, development for PAT to control glucose at a particular level to limit glycation, which impacts the bioavailability. So, yep, the new inline probe was identified for controlling the glucose, provides a faster alternative to existing techniques, obviously published in an article. From a formulation perspective, this was developed and optimized for drug candidate under GLP conditions. Proof of concept demonstrated for the immunoassay for the inline probe. Currently, the PAT probe has been built to be used for drug candidates, so the design for prototype 1 was complete. From a prototype 2, currently used for development of new clinical trial materials. From a BRL6 perspective, development with a drug candidate, this is the pilot operations 1. This is where the new technique currently is used in development of new drug candidate for clinical trials. Bridging studies underway to demonstrate the PAT probe versus the inline method that's currently ongoing. From a GMP ready, system ready built perspective, this is the BRL7. Since this is a PAT probe, instruments qualification underway to demonstrate the control during GMP make. And method currently at BRL6 and progressing to achieve BRL7 as listed out in Appendix A of the article. So this is again, similar concept. I want to pull out some of the questions that this team was able to kind of address. They were addressing the production scale tech availability. They were working on supply chain establishment for the longer term to develop the PAT and deliver the PAT across the spectrum. From a quality readiness perspective, the team was working on establishing the QRM, quality risk management plan, and change control process as identified by ICH Q9 and Q10. And obviously the commercial cost model established was obviously established earlier on, but refined over the time with the GMP system built. And some of the other aspects that were identified were risk identification by key stakeholders. This is where you would want to make sure you have a proper plan for stakeholder management. And risk are identified throughout the process and are addressed before you reach to the next step. Team was working on optimizing manufacturing plans for commercial units. So for more information on this document, you can visit our website. You can download the publication that has been published as an open access in biotechnology and bioengineering. And this will detail in a little bit more information about each of the 9 subcategories, which are listed out here, and the phases of development. And coming from a CMC background, we are basically developing a product here. We are developing a product from a technology perspective to be implemented in biopharmaceutical manufacturing. So that was the concept. So what does BRL provide? The framework provides for performing assessment of new technologies or process for technology adoption. And one of the key items basically for technology adoption is this quality readiness section, which basically is a key criteria which differentiates us in a pharmaceutical industry settings. It's an assessment toolkit with the end in mind. So the end in mind, obviously is the commercialization of technology or the method. And the commercialization can have different aspects. Some of the commercialization basically is to use in a commercial product of biopharmaceutical, or some could be development for R&D use only. So it's basically thinking about what is the end in mind that we're using this framework for. This framework will help you identify the risk and gap analysis during the development of the technology. It will show you a roadmap for that progress over time has been made during the technology development or method development. It also basically is used for portfolio management for tech implementation and identifying which technologies are at advanced phases, which are at earlier phases of development. With that, I'd like to conclude by saying that please provide your feedback on the BRL website, or you can also email at brl@nimbl.org. We have published this in Biotech and Bioengineering journal. Please, this is open for comments up to 1st of March of next year. And with that, I'll pause for any questions by now. Chris, over to you.
All right, thank you, Sandeep. And thank you for everyone who've been populating some questions in here. We have plenty. Some are shorter and some are longer. And I think some of them came in along the way, so maybe you covered bits and pieces, but I think they'll still be worth a good discussion. So I'm going to start with a simple one. Well, maybe simple. I'm going to start with a short one. How is the BRL assessment different from the commonly used TRL system?
Thank you for those questions. So we have used TRL and MRL framework as a starting point. So we definitely are building on that. The difference that you will notice in the BRL framework is basically it's industry specific to biopharmaceutical industry. And we have tried to address things like quality management systems, change controls, ICH guidelines, other regulatory guidelines that you need to be considerate of when you're developing a technology or you're developing a method or a new assay to be implemented for biopharmaceutical products. Because at some point in time, you will have to address those questions to be included as part of the manufacturing paradigm into your BLA files.
Okay.
Next.
All right, so let's pick a different one. So here's an interesting one. I'm going to paraphrase because there are a couple that are in sort of the similar space. But when you think about biopharmaceutical manufacturing technologies, it could be that the value proposition of the therapeutic itself could be an overriding factor in whether it has advanced to commercialization rather than the maturation of the manufacturing technology. An example might be cell-based therapies. So the question is, can or would or should the BRL assessment take this into account? And maybe that's something for consideration for version 2.0.
That's a great question. Thank you for that question. And I struggled myself throughout my CMC career to address those questions, right? So when do you implement a technology? I think it's a case-by-case study, right? And looking at cell therapy or gene therapy, which are new modalities, and they're fast, rapid pace at some times, and waiting on clinical trials for the others. There definitely is an insertion point. And the reason for the BRL framework is actually to give you all the challenges or questions that we would need to address at some point in time. Not all of them would be applicable to whatever you're developing, but hopefully majority of them would enable you to think when you need to address those questions. So for instance, you're developing a cell therapy, you're going from phase 1 to phase 3, and this is not more about developing a CMC paradigm, but at some point in time, when you're implementing the new technology or innovations into the clinical trials, you will have to bridge something with the existing one. So I think it provides you a framework on when it's appropriate for you to introduce a technology or the assay into your development timelines. And it's a case by case. There's no right or wrong rule from that perspective, but it basically provides a general guidance to you in terms of what would be required if we have to implement this for a commercial product eventually. So obviously, the modalities take the helm compared to the technology development at some times.
So maybe related, I'm going to kind of combine a couple of questions here into a similar vein, and I think you touched on some of it, but I want to give you a chance to share your thinking. So when you think about a small company that has a technology that might be considered for implementation within a biopharmaceutical manufacturing process, including things that might be around quality control. So maybe this is a sensor, for example. And so it's really, it's a couple of questions that kind of come together to ask the question of, should the BRL be considered from both the readiness of the instrument and the demonstration of readiness within the manufacturing process? And then if you think about quality, there's also a question of at what point do regulatory standard requirements, ICH, et cetera, come into the evaluation of performance? And then there's, of course, any comparability to existing technology. It's sort of merging a few different thoughts, but I think they kind of are in the same space for you.
Okay, so I'll go question by question. There are 3 subparts to that question, and thanks for that question, right? So the reason we developed this BRL framework is actually exactly for that reason. We have innovators, we have developers, and we have implementers. So innovators could be an academic lab or a small to medium manufacturers, and developers could be a small to medium manufacturers or a large biopharma, or implementers are biopharmaceutical manufacturers, right? Could be CDMO or a parent company, right? So the reason for this framework is actually to collaborate. The reason to actually communicate, to share a common understanding. What is required to understand upfront the different aspects that would need to go into the design and development of the technology per se. So it's to develop that framework, right? And to have that open communication, because most of the time, the development of technology takes place in a silo, And when we go to basically implement our technology or say advertise our technology for a biopharmaceutical product, we always struggle to overcome the burden. The burden basically to overcome essentially implementing that technology for a clinical manufacturer. So it's basically a framework to get over that hurdle. To have that communication, to understand what is required from stakeholder perspective, and then start to incorporate some of the design aspects into the technology perspective. In terms of the regulatory question that was asked, when to address some of the ICH guidelines, things like that. I think the guidelines are pretty well written out. It's up to the interpretation of the developer as well as the implementer. I think it needs to have an open communication on when they would like to incorporate some of the design aspects for that technology, right? As per the guidance. Sooner the better. Later, it's going to just delay the development of the technology. I mean, it could be validation, verification, things like that, right? So there are different aspects that need to come in place. Again, collaboration is a key word here. Basically, understanding the guidelines is a key word, and basically getting in touch with the implementer earlier on, so you understand their pain points on why they can't or cannot implement your technology, for instance. And I forgot the last question, Chris.
No, I think you merged a lot of that together, and then it would roll over into additional questions that are sort of really focused on that higher BRL level, that 6 into 7, which can you comment on maybe challenges that may be there for actually implementing this in the environment where you have a real drug candidate, and is that something where NIIMBL might get involved or should not be involved, right? If it's those higher levels, as well as then when you get into that higher level where you might be using pivotal lots, then it may not work well for evaluating new technologies that are being applied to licensed products. So there might even be just a question. It might be that, like to your point of vocabulary, do we have a shared understanding? Do we have the right wording in order to help people navigate that space?
That's a great point. Absolutely. Those are great questions, basically. So I'll rephrase my answer for those. In terms of BRL6, right? So this is where you're introducing your technology. You've done all the POC work. You basically demonstrated your technology works on a pilot scale using a feedstock, representative feedstock, things like that, right? Now you want to introduce that in a clinical trial. So there are aspects, GMP aspects that will come into consideration. And we need to have that open dialogue with the implementer on how and when to achieve this. What are the design requirements, right? So it's usually around the design requirements that are the key challenges. What are the paperwork? What are the validation aspects and verification aspects? Those are required. In going from BRL 6 to 7, where you're talking about now you have demonstrated on in the clinical trials, it could be early phase clinical trials, and you want to demonstrate that technology into the late phase clinical trials or pivotal makes or second generation processes, right? And this is where you have some of the background built up from your clinical trial BRL6 concepts already. And you're trying to demonstrate the robustness for the technology. And those are usually questions around robustness, what questions you're trying to answer using that technology, whether it's a model control aspects, quality control. So those are things that are laid out in the framework. Now, obviously, the framework is not extensive. Hence, we are asking for your open comment. Hence, this BRL framework is open for comments up to March of next year. We want to make it an exhaustive kind of framework so that we can think about all the nuances that are probably missed in the version 1.0. And they'll definitely, we are hoping there'll be a version 2.0 as we can combine all the comments and suggestions, recommendations that we receive.
All right, thank you, Cindy. Let me take, there's some thoughts here that are going a little bit of a different direction. So this is a question around that with the work that's also being shared separately, I think within this webinar series, that NIIMBL is developing platforms of a sort and test beds that include things like protein manufacturing or viral vectors or others. Could those be used as an ongoing resource for de-risking of new technologies and demonstrating them in relation to current methods? And that may also then tie in with the question of the relevant material, because some of those would be not actually clinical candidates, but they would be industry relevant.
Yeah, I think the question there more is GMP-like versus GMP. So I think what we are developing in NIIMBL headquarters is basically GMP-like. Yes, though we are developing test beds, they can certainly be used. Depends upon what we would like to demonstrate in those test beds and how we could use it. Obviously, there needs an open dialogue again, developer versus the implementer. In this case, the NIIMBL headquarters is the implementer and developer is the technology developer. So yes, those questions can be answered, but there's a key difference between BRL 6 and BRL 7, and that it is for a purpose. The purpose is your unit in terms of PAT has now been qualified, validated, or things, whatever guideline requirement is to be used for a pivotal make. And I don't think we have plans in NIIMBL to make anything pivotal makes in our facilities. That's an uphill task there. But I can speak for the future, but currently we probably don't. And we need to look at where the pivotal make needs to be done. In most of the industries, pivotal makes are usually done where you launch the drug from. And it could be a different case from different modalities, right? So it's quite open that's why I want to separate out the CMC questions to technology development questions. Technology development, the framework is to support the CMC filing eventually, right? So you got to think about what would be required when you actually introduce this technology into the pivotal make. And there is a lot of validation work that needs to go into it, that needs to be basically pulled up and completed prior to be implementing into the pivotal makes, because your data relies on it very heavily.
All right. So I'm seeing a couple others here. I'm not going to ask you to restate some things you said. The ones that are sort of touching on the level of when one would engage with regulatory agencies, and we certainly are not in a position to speak for the regulatory agencies. So that, as you said, that would need to be a decision by the individual innovator. But there is also one that is specifically calling out from a small instrument vendor perspective. They note that a number of the activities and milestones are quite heavily dependent on partnering with a biomanufacturer. And so an ask they have, and I can probably help to give the answer to this one, is, will NIIMBL assist in creating these partnerships? If you are a member of NIIMBL, that is part of our role. And so we won't address how you do that in this webinar, but you can reach out to your partnership manager. I thought I saw Nicole Murley was on the call, or others that within our business development group, and we can help with that. And then is it a related question from, I think, the same perspective, is that do we think that it's likely that a given BRL level will be recognized by some biomanufacturers for their drug products in this particular example, but not by others? Or only for certain drug products, but not for other drug products, for example. Which it's a question of general recognition, which I think is a theme that you were trying to get to, but I wanted to give you a chance to comment on that.
I'll respond to the second question. Thanks, Chris, for taking the first one. Yes, absolutely. We would like this BRL framework to be adopted throughout the biopharma industry, right? The more we adopt, the more we have a common vocabulary, a shared vocabulary, the more we have a common understanding of what it takes to develop the technology. Because at the end of the day, any technology developer has the end in mind, which is technology implementation, right? Adoption. How does it get it? So getting over the hurdle is a key part. Hence, we were kind of so much interested personally in developing this BRL framework. We hope that all industry throughout the biopharma adopts this framework, and we would like openly, hence this webinar is open for members and non-members. Please send in your comments where you find holes or gaps in the framework, so we can kind of publish a version 2.0. But at the end of the day, we would like to create a common harmony, a common kind of vocabulary to understand and talk and dialogue, have a dialogue with our developers, our innovators, our implementers on where your technology stands. So if somebody says my BRL is at 5, everybody knows what they're talking about, where their technology stands. Or somebody says my technology is at BRL 8. We know where they stand, right? They have already established a commercial stage technology development, basically, process. So we would certainly hope this harmonizes across our industry, and we have more adopters, and we have more implementers, and we welcome your feedbacks. We welcome your feedback, because this is where we can kind of create a harmony in providing a framework.
Right, to that one, I think I'll grab one last one that's in here. which was a little bit earlier in when it showed up, but I think where the flow is working with this quite well now with how you're going with the Q&A, is that at least one of the folks was able to share for us that the way they approach the problem is to come up with an equivalent of a target product profile for a new technology. Sort of, I guess, like you might think about the product profile for the new product. And then try and align, internally with any partners on what they see as the key parameters or the maybe obstacles that need to be overcome in order to meet those expectations. So I don't think it's to me, it isn't that different from what you've been pointing out, but I think they're asking really, is there a way to put that, to have that sort of context in there as part of the format, because that may be the way others approach the problem. As you said, ending goal, working backwards, right? And so, and that would maybe just be, do we need to make sure that we've captured all that through the feedback that we get through the email address or the website? But they wanted to make sure to hear your thoughts and comments.
Yeah, certainly, and I appreciate that comment. So it's combining the CMC aspects for biopharmaceutical products versus technology product. And I just want to basically, highlight that we can certainly, we are intermingling the space anyways, because we are bringing in the aspects of technology development and the ICH guidelines into it, right? When QTPP or quality target profile or product profile QTPP comes in play, we're targeting the CQAs and aspects of those, right? So yeah, we can definitely bring those aspects, and we welcome your comments. So please share, provide your feedback in the email so we can elaborate that question a little bit more, so we can understand exactly how we can incorporate it into the framework. But we certainly would like to incorporate the framework, anything that's relevant to technology development, because at the end of the day, we would like this framework to be kind of your roadmap for technology adoption. And I think that's a key challenge, and that's a key challenge and key ask for us as well as industry as a whole.
Yeah, and to help to drive those conversations, whether NIIMBL is involved in those conversations or not, is not that isn't
correct.
That's not the end result that's being sought. It's more to help facilitate.
That is correct. Yeah.
All right, I think we've covered all the great set of questions. Thank you to all who took the time both to listen in and go through the presentation with us, but also for taking the time to put questions in the chat. Again, this one's being recorded, but we may also, if there are any that maybe popped in a little late that we find later that we didn't cover, we'll make sure to cover that through maybe a short FAQ that could just be written and posted along with these slides. So I think there might be a few last closing slides. Is that right?
That is correct, Chris. So I'll go through the closing slides. How do you follow up on NIIMBL projects, right? So basically, all you got to do is you got to follow a project on our NIIMBL website. You'll receive an email alert when you click on follow project, which has an update. The email will have a link to the project. You can click on the link and you get an email. You directly go to that web page. So it's very simple. Basically, you can click on a number of projects that we have ongoing at NIIMBL. We can just say click to follow, and you automatically get an email update whenever there's an update on the project. Lastly, but not least, on closing, I want to remind folks, thank you for again attending. You'll receive a survey link shortly after this webinar is complete. Please complete the survey so you get immediate access to the webinar recording. Want to remind, we have a few other webinars and conferences coming up in NIIMBL. Some of the dates we have on February 7th, we have virtual internship co-op and early talent career fair. So employer registration now is open. In Q1 of next year, we are doing a NIIMBL program web series, which will consist of PI, process intensification, vaccine manufacturing, and NMAP. In the same Q1 of next year, we are also conducting a big data workshop, which will have both the components for members and non-members. And we want to remind folks that our NIIMBL national meeting for next year is scheduled for June 27th to 29th. And with that, I thank you and hope this is a roadmap, a journey, a train that we can all follow through. Thank you.